First of all, thanks to everybody for being here. I'd like to thank the press and especially Corvelva. We will introduce today's guests, but first I'd like to say a few words regarding the spirit in which this conference was organized. My name is Sarah Cunyal, member of the Five Stars Movement, as well as member of the Congress of Deputies Agriculture Committee. I come from the city of Vicenza in the Veneto area of Italy. We are here as deputies because we are required to represent the citizens. The parliament where we sit has to return to being the House of Citizens, and our primary duty and responsibility is to make their voices heard. We are here today as spokespersons in this place that, in my opinion, must return to being representative of all citizens, a place where their requests and concerns will be heard and embraced. I'd like to mention that at the moment we're discussing and voting on a law on the topic of popular referendum in Congress. The purpose of this law would be to turn Congress into an instrument of direct and participatory democracy. Therefore, this is the spirit in which we have organized this conference, in response to the tactics of division, discreditation, and use of derogatory labels that the media and government have employed, which unfortunately embrace the use of harmful propaganda over the honoring of democratic principles. Thank you again, everybody, and I'll let Corvelva speak. Good morning, everybody. My name is Nassim Langrudi, and I'm a member of the Corvelva Association, which has been fighting for freedom of choice on vaccination since 1993. Here with me is Andrea Rinaldelli, father of the late Francesco Rinaldelli, a former soldier in the Italian army whose story is related to the Parliamentary Commission on Depleted Uranium, which looked at the administration of vaccines in the Italian military. He will have a few words to say about this later on. Also with me is Dr. Loretta Bolgan, PhD in pharmaceutical science, who's been handling the scientific aspect of the vaccine analysis commissioned by Corvelva. We are here today to inform as many people as possible of some important steps that have been taken by everyday citizens who are democratically demanding to be heard and answered in a manner that has not occurred to date. Corvelva took on the responsibility of acting on some findings, which the Parliamentary Commission on Depleted Uranium had already highlighted as relevant in its final report concerning vaccinations as a possible health risk for the members of the Italian military. Ivan Catalano, the Vice President of the Parliamentary Commission, was also supposed to be here with us today, but unfortunately, for logistical reasons, he was not able to make it. Later on, I will read the speech that he had prepared for us. In addition to this, we urgently need to report the results of the analysis we have done on a few different vaccines. Our association has commissioned 2 independent laboratories to carry out a chemical and biological analysis of the vaccines we are testing. This research was also part of the recommendations made by the Parliamentary Commission on Depleted Uranium. Specifically, the committee indicated the necessity that an analysis be done on samples of vaccines, and this is exactly what we, the citizens, are doing. We are taking action on what government has not pursued. These results will be presented by Dr. Loretta Bolgan, consultant to the Parliamentary Commission. We're demanding that these data, which outline a potential risk for health, are taken into consideration, because many of the vaccines whose contents we are analyzing are currently in use on the Italian pediatric population and are among the vaccines made mandatory by Law 119/2017. The government is imposing the administration of pharmaceutical products that, when analyzed, show to be potentially non-compliant, thus affecting these products both in terms of effectiveness and safety. With this being said, we are demanding that appropriate precautions be taken, because we're not just talking about the health of our own children, but of all Italian children, obviously in particular those who are on the receiving end of the recommended vaccination schedule. Sadly, we have had to acknowledge that our requests have fallen on deaf ears, and in particular, we want to point out that the Minister of Health has not yet given us any kind of feedback regarding our communications, nor to the notifications that we sent her as soon as we received the vaccine analysis reports from the laboratories. Yet, as you will hear, these are not minor findings that our testing has uncovered, but instead findings that cast major doubt onto both the quality of the vaccines and the controls used to test the safety of the finished products. In fact, today we are asking, where are the safety tests that have been carried out in recent years? A discussion is necessary in order to verify what we are now bringing to everybody's attention. The regulatory authorities have given partial answers to just some of our requests and notifications, mainly responding to our questions about testing methods. Today, however, we are informing you all of some observations that raised questions. These same questions have been passed on to ISS, EMA, and AIFA, but have remained largely unanswered. When faced with questions about noncompliance regarding what is stated on the vaccine data sheets and reporting potentially harmful elements, the answers we received defend the formal authorization and control systems. We expected them to engage in prompt interventions and analysis in order to actually verify what we are exposing here, but this is not happening. We would also like to say, however, that the divide between government and citizens, between institutions and civil society, has been evident on many other occasions and has become exacerbated in recent months, particularly around this crucial issue of vaccination. In 2018, a number of citizens set up a promoter committee, the Committee for Freedom of Choice, to draft a citizens' initiative law, as provided by the Italian Constitution. This bill was brought to all Italian town councils, and in just 4 weeks, in the middle of summer 2018, more than 100,000 citizens went to their town halls to sign and bring their voice to the political institutions of this country. The bill, which is called Suspension of the Compulsory Vaccination for Developmental Age, calls for a nationwide adoption of no mandatory vaccines, modeling the policy that's been in place in the region of Veneto for almost 10 years now. On September 20th, 2018, the Promoter Committee filed 75,000 signatures at the Chamber of Deputies. This was on another occasion on which the government decided to ignore the demands of the citizens, who wanted to bring politicians back into dialogue with citizens and restore the right of citizens to take part in legislative activity, as is guaranteed by the Constitution. On the contrary, we find ourselves living in a climate of persistent tension on the issue of vaccination, with positions becoming increasingly harsh and despotic, informed by a dogmatic rather than a scientific approach. Now, data cannot be anti-vax or pro-vax. Data are data. We are demanding a scientific comparison of the data from analysis of vaccines be done in an absolutely scientific way. Instead, the focus keeps being put on the existence of two supposedly opposing factions, which have actually been invented by the media, and which didn't even have names before 2016. The issue of mandatory vaccination is still very much a part of the political landscape, now that draft law DDL 770 was filed on August 2018 and signed by representatives of the Five Star Movement and Lega. This draft law is currently being examined by the 12th Committee on Hygiene and Health of the Senate. To date, the Promoter Committee has not yet been granted an audience, and these citizens are once again being ignored. We are demanding to be respected as citizens, to be enabled to exercise our fundamental constitutional rights, and today, more than ever, facing the data that will be presented to you today, we believe it's necessary to get real answers supported by data, not only supported by words. We hear it said that vaccines are clean, safe, and effective, but the only tests we have before us at the moment are those we are paying for. Where is the proof of safety and effectiveness, and where are the analyses showing that these data are possibly wrong? I will now turn things over to Andrea Rinaldelli, whose story, as I mentioned, has been deeply explored during the Parliamentary Commission on Depleted Uranium, and who is still waiting for answers regarding the death of his son, Francesco. Hello everyone, my name is Andrea Rinaldelli, father of the late Francesco Rinaldelli. My son died of an illness on March 16th, 2008. To keep things simple, I would like to focus on just one matter. My fight lasted 14 years, and what has happened is unacceptable and ridiculous. After the death of my son, I wrote a letter to the then President of the Republic, Giorgio Napolitano. His letter and answer included traditional greetings and condolences, but also an acknowledgement that I will read. At the end of 2007, the CPCM Defense Military Disease Prevention Committee was established, composed of scientifically competent researchers from the Ministry of Health, Ministry of Defense, Ministry of Research, and Parliamentary Inquiry, presided over by Senator Lilia Menapase. The tasks of the CPCM included analysis of vaccines. Keep in mind, this is a letter I received in 2009. Following this exciting initiative from the Minister of Defense, a second committee was established. The first one was presided over by Sergio Costa with the aim of investigating the content of vaccines. As you can all imagine, nothing ended up happening. Following this inquiry, a series of legislative actions were laid out in order to examine the military situation related to the administration of vaccines. At the end of the second commission, a third was established, and it just ended in 2017. Like the two previous ones, the third commission had the same responsibility to investigate the contents of vaccines. I think it's hilarious that I must be grateful to Corvelva, who finally carried out the work of the commission. The government made a commitment, as evidenced by the letter that the President of the Republic, Giorgio Napolitano, wrote to me, to send these analyses of what's in the vaccines, so that I could take judicial action, analyses that could lead me to the truth and justice for my son. This hasn't happened yet because in all judicial courts they deny the correlation and they state that there is no scientific data to prove it. So I'd like to ask you, does this sound fair to you? I'm asking this question to you all. It's a personal thought that I'm expressing because I think that the servicemen and servicewomen, with the sacrifices that they make, have made it possible for Italy to brag to all the world about their competence. But I don't think that the institutions really honor them by investigating the true origin of their illnesses and giving them appropriate compensation, because it would only be fair that they receive compensation. I want to explain to you all my doubts and also make a comment about CPCM. CPCM is comprised of several scientists: Lieutenant Colonel Don Vito, Professor Gobin, Dr. Antonietta Gatti, Dr. Gennaro Valerio, Professor Lauro Renato, Professor Lenzi Andrea, Administrator Martina Di Vincenzo, Professor Muti Paola, Professor Razzi Guido, Dr. Salmaso Stefania, and Professor Zucchetti Massimo. On the first term of the mandate, Professor Lenzi was replaced with, what was his name? Just 1 minute. Luca Pani. Do you all remember who Luca Pani was? He was the director of AIFA, the National Medicines Agency. Soon after that, he was moved away because his situation was somewhat embarrassing for the Minister of Health. How is it possible for a commission set up by these people to come to acceptable and clear conclusions on this matter? We all know that Dr. Salmaso, on more than a few occasions, pointed out her doubts about us and the anti-vaxxers. How can she, who clearly already has strong opinions regarding those who do not vaccinate, represent justice and equality on this matter? It's the same thing with General D'Amelio. He is a very important piece of the puzzle, one of the most influential Italian experts on vaccines. He is the creator of this project, the leader of CPCM. Where did all the data end up? I want to point out that CPCM had 5 years to complete their inquiry, but that in all that time they never submitted a single study, not even one, that made sense. They were given grants amounting to something like €2,830,000. So why is it that they didn't set aside any of this money, let's say €100,000, to analyze the content of the vaccines that the soldiers were being given? I think this is an important question to think about. I've tried to summarize as we have limited time, and it's very important to hear what Dr. Bolgan says about the analysis findings, because she was one of the representatives at the last commission. Thank you. I'll just read the speech prepared by Ivan Catalano, who, as I said earlier, couldn't join us today. Today's press conference, almost a year after the final report of the Parliamentary Commission on Uranium and Vaccines was published on February 7, 2018, not only confirms but also updates these results, thanks to a dedicated faction of the Italian scientific community. In fact, with their analysis, Corvelva has confirmed the problems with the content of vaccines that were alluded to during the investigations of previous committees. Based on these data, we must start a reflection on the risks of mandatory vaccination, correctly guiding discussion of Bill 770. The risks are the same whether the people being vaccinated are military or civilian. The risks are due both to the vaccine inoculation procedure and to the composition of the vaccines available on the market. The precautionary principle is the basis of a careful assessment of risks with the aim of protecting not only the community, but also the rights of individuals to live without serious and disabling adverse reactions. The Constitution reminds us of this in Article 32, paragraph 2, so let's not forget it. Authorities must conduct studies to investigate rather than to discredit. We seem to have gone back in time in terms of human rights and so-called civilized society. Instead of creating a culture of persecution, a sense of social division, and a completely useless hysterical fear, authorities must work to protect the individuals as well as collective health. I am hereby announcing that I've sent a scientific article concerning the epidemiological analysis of the data published by the Commission on the Health of the Military to an epidemiology journal, which has evaluated the study. They have informed me that they will consider it valid for publication once I've accepted the proposed changes. The results confirm once again what the Commission has published. Once the study is available online, I will request an audience from the Defense Commission to present it and file it. I will now let Dr. Bolgan speak. Here I will do a short review of the major findings we obtained with the first phase of screening on the analysis of some vaccine batches that are currently on the market. I'd like to make it clear that AIFA, the Italian Medicines Agency, as part of the Parliamentary Commission of Inquiry on Depleted Uranium, had stated and confirmed that the military vaccines are the same as the civilian ones. Therefore, we are expecting that the issues that arose from these studies can be considered valid for both the military and civilian vaccines. What we've embarked on here is an honest evaluation of the commonly stated so-called fact that vaccines are clean. So we want to understand what the word clean means, because for an ordinary person not in the field, clean means that a vaccine contains the active ingredient and the excipients and nothing else. In pharmaceutical terms, this isn't the case because contaminations and impurities can exist, but when they exist, they must be well defined in their toxicity and must have a specific definition that can confirm that even if they are present, they are not harmful. We wanted to test the quality of the compounds, so the first study that we have conducted is about compliance, meaning that our goal was to verify the vaccine components as declared in the datasheet or the insert. So we analyzed the active ingredients and the excipients. After that, we did a screening called first level screening, a first study just to see what the vaccines contain. This involved an identification of the components, whether they were chemical substances, proteins, or DNA material. The last one I just mentioned, DNA material, shouldn't be present in the vaccine, and if it is, it must be regulated in a very rigorous way. Also, it must be specified on the datasheet as residual matter. Normally on the datasheet, you find residuals such as formaldehyde and antibiotics that are actually tested by the producers. But what we have found in the vaccines that we've tested is slightly different from what is declared on the datasheets. We have completed the screening phase for the first 4 analyzed vaccines, and we will talk about them shortly. We've already begun the second phase, which we are calling second level analysis. I have to say that we have already pre-published some results, so we haven't come to the peer-reviewed publication yet, and this fact created some commotion within the scientific community. I will explain why this is, and once we have obtained the results of the second level analysis, we will proceed with the data qualification, engaging with additional laboratories to retest our findings by certain parameters and verify the identities of the compounds we have found in the vaccines. We are hoping to have the chance to cross-validate the results in phase 4 of our analysis, which we are aiming to complete by March. The scientific community has been divided over the fact that we publish the data without a peer review. When we were discussing how to present the findings, it appeared necessary to answer this question. Should we tell people now, or should we wait a couple of years for the data to be published with peer review? As a parent, because I am a parent, if I have to vaccinate my child, or if I decide to vaccinate my child, I would very much like to know right now, because I would want to be able to apply the precautionary principle and weigh the risks, because this data raises doubts, as Nassim said, about the vaccine's safety and effectiveness. These are doubts that urgently need resolving because this research takes on a double meaning. It's not just scientific research that we're doing here, but something with social relevance and impact. Because if we would have waited another couple of years and only shared the data once it had undergone peer review, meanwhile, thousands or maybe millions of babies would have been vaccinated without knowing of a potential risk. And this, in my opinion, must be taken into full consideration by a regulatory agency and the Minister of Health. Therefore, we're making the results available now so that further investigation can be done immediately. We're not asking the Minister of Health to reproduce the analysis we've done, as we assume they have already done the analysis. And also because the lots of these vaccines have already been issued. Rather, we want to see what data they already have. We want to understand why our results are different from theirs. This is what we're asking for at this precise moment. So we have used two kinds of technology, and we haven't disclosed the names of the laboratories that we used because, as I said, we're in a phase of pre-publication and of data submission to scientific journals. The scientists chose to wait until at least the pre-publication of the study before openly discussing their analysis methods. So I believe in a short while we'll be able to make public the methods, names of laboratories, and scientists with their permission. I'm talking about laboratories that are working on behalf of third parties. Their work is focused on analyzing complex matrices with the latest advanced technology, and they have all the appropriate certifications to perform these kinds of studies. Again, the first screening level has been done to identify the unknown vaccine components, and we cannot apply the standard validated methods to analyzing the unknown vaccine components that were found, because the standard validated methods can only be applied to analyzing known components, which is a normal process for the manufacturers of the vaccines. So the phase we are in is one of research and development, which will need to go beyond what the manufacturing industries currently do. The first type of screening concerns genetic material. So we have looked for DNA, RNA, and viruses that can accidentally contaminate a vaccine. We have used a system called next generation sequencing that until 5 years ago wasn't even well established as an analysis method. Now, however, it is considered a routine analysis method that is also available privately. Another important method is mass spectrometry, and with this technology we have been able to investigate the chemical and proteic parts in the vaccines. Also, this technology is currently used for forensic analysis, and this specific technology has been acquired by the FDA for the analysis of contaminations in complex matrices. So we are talking about a very reliable, accurate technology. We've already finalized the analysis on 4 vaccines. We've analyzed more, but I will just talk about the ones we've completed as part of the first screening level. I think they're well known. We have Infanrix Hexa, a hexavalent vaccine produced by GlaxoSmithKline, consisting of a powder and a solution, which can be useful to keep in mind. We also have Priorix Tetra, which is a live attenuated virus vaccine, measles, mumps, rubella, and varicella. Hexion is another hexavalent vaccine produced by Sanofi, and this is a pre-prepared suspension solution. And we have Gardasil 9, which is the anti-papilloma virus vaccine, which contains 9 types of strains of the papilloma virus, the ones that are mentioned below. I won't mention them all now. So on the Infanrix Hexa datasheet, it's specified that the vaccine contains traces of formaldehyde, as well as neomycin and polymyxin, the 2 antibiotics in use in the finished product, which can cause allergy issues. So these ones have been declared on the product datasheet. According to the first results regarding DNA in Infanrix Hexa, the genomic part, we weren't able to find the poliovirus as reported on the datasheet. And EMA has responded to us on this issue, saying that it's fine, we didn't find any because the formaldehyde treatment that is used to deactivate the diphtheria, tetanus, and pertussis also destroys the poliovirus's genetic material. So what remains is a protein called antigen D that is able to create the antibodies. We've also found traces of monkey DNA originating from the Vero cell line, which are immortalized and potentially carcinogenic. We performed a high-level chemical analysis of the residual growth medium contaminants, beyond those which are reported on the vaccine datasheets, and found that these elements are proper components of the vaccines, which should not be classified as a residual. They need to be tested for their toxicity and reported in the datasheet. We have found 65 signals that are not expected vaccine components, only 35% of which we were able to identify, while the identity of the remaining 65% are unknown, and we have also 8 toxins in the vaccine. Priorix tetra has been produced on 2 cellular lines, a chicken embryo cell line and diploid human cell MRC5 line, which originated in a male fetus that was aborted at 14 weeks of age in 1966, an old line, which can also present several issues, such as issues with growth. We had difficulties growing it to see if it belonged to this line of cells, and the metagenomic analysis has brought up several issues on this specific vaccine. First of all, we have found a relevant quantity of fetal DNA that was coming from a complete genome, so we have an entire human genome in this vaccine, which can be classified as a vaccine component. We didn't find the rubella. We performed an extremely deep sequencing with 260 million sequences analyzed, and we found only 114 of rubella. As a result of this study, we have found 4 contaminant viruses: endogenous retrovirus, leukemia virus, equine anemia, and avian leukemia virus. These viruses are widely studied in human anatomy, and they can bond with human DNA. This being a vaccine containing an attenuated virus, it would be appropriate to understand if these ones are deactivated or if they have been analyzed, because they present a hazard profile and mostly the quantity far exceeds the quantity of rubella that's present in the vaccine. This poses a major question because it means that rubella isn't there after all, and from an immunological point of view, it is not present in high enough concentrations to stimulate the production of antibodies. So if rubella can produce antibodies in such a low concentration, perhaps the other 4 contaminant viruses can as well. As for the chemical and protein analysis, we have the same situation as with Infanrix hexa, with a far higher number of contaminating chemicals than the vaccine should contain. We also found 2 allergenic proteins able to produce antibodies, potentially dangerous, and chemical substances deriving from, as in the Infanrix hexa, contamination of raw materials. We also found herbicide, miticides, substances found in chicken foods, which they get from the eggs to grow the viruses, but also cross-contaminations from different production lines. This was a very big surprise, and the quantity is not minor. We will verify these substances to make sure of what they are by a certified standard control analysis. Hexion is the other vaccine that we compared with Infanrix hexa. What happened when we analyzed Infanrix hexa was this: we couldn't find the antigens that were supposed to be characterized as proteins. What this means is that when we test, we need to recognize a series of specific characteristics in order to identify something as a protein. It has to be soluble and digestible by trypsin, and we have to be able to sequence it and have the amino acids. With Infanrix hexa, this wasn't possible to identify, so we didn't find any. Instead, we found a macromolecule, indigestible and insoluble. And now the question is, can this macromolecule create protective antibodies or not? Thus far, we haven't got an answer, but when we analyzed Hexion, we found 3 out of 6 antigens, and we did find the toxoid antigens, diphtheria, tetanus, and pertussis, using the same method. We were not able to identify the other 3 antigens. According to what we can find in the scientific literature, and there are lots of studies on this issue, it seems that the use of formaldehyde and aluminum adjuvant in vaccines is forming chemical bonds that structurally modify these proteins and make them incompatible with intracellular enzymes, rendering them impossible for the body to break down. They can also roam around the body, and they remain there for a long time because they are very difficult to excrete. So once again, the question is, if the proteins have changed their actual form, do they still create protective antibodies? This is why these answers are urgently needed. Regarding the metagenomic analysis of Hexion, we have found, unlike in the case of Gardasil, a lot of DNA contamination that shouldn't be there. We have adventitious viruses and monkey DNA coming from Vero cell lines, which is not supposed to be there because it's a carcinogenic form of DNA as it can bond with human DNA. Here also we have 260 signals, 16 components that are toxic chemicals, and only 30% of those could be identified. The last vaccine, which is very important that we discussed due to the worrying outcome of our research, is Gardasil 9. In terms of the metagenomic analysis, we've uncovered a situation that's already been discovered by other researchers. We found human DNA and mouse DNA, which we suppose is a raw contamination because we don't understand where it came from. We found adventitious viruses and papillomavirus DNA fragments that, according to scientific literature, are reported as dangerous because in bonding with aluminum, they can travel with macrophages. These have been found in brain tissue and other parts of bodies of girls who died after vaccination. We also found retroviruses, leukemia virus, viruses that bond with DNA, and also synthetic constructs, scraps of synthetic DNA that are currently used for genetic recombining and that bond with aluminum and remain not degradable and not soluble. They can wander in the body, potentially doing damage that's currently unknown. In terms of the chemical analysis, we found 338 signals, only 22 of which have been identified, so we have some information about these, but the remaining 78%, no, because we don't know what they are. We didn't find 2 of 9 strains of antigens that were supposed to be there, so to be honest, it seems to be Gardasil 7 and not Gardasil 9. So this is the situation, briefly illustrated regarding our work. The issues discovered with the vaccines, which had already been anticipated, mean that we have problems with efficacy because the antigen hasn't been found. We have the presence of chemical and protein contamination that cannot be defined as residual because it exists in significant quantities. And we also have virus and contaminant DNA and RNA that can integrate with human DNA or cause autoimmune diseases, which has already been documented in the scientific literature. All these problems raise questions about the quality of these vaccines being not 100% guaranteed, and if we can't guarantee the quality, we cannot guarantee the efficacy nor the safety of these vaccines. Thank you, Dr. Bulgan. I don't know how much time we have left, but if anyone has questions, please go ahead.